Hi, so along with running Galaxy Nutrition, I work here at a molecular oncogenetics laboratory. And so today we're going to be showing you how to do a fecal soil extraction with the Zymo research kit. And this is catalog number D6012. So normally in this procedure, you're supposed to put your soil sample in a bashing bead buffer. Which is this right here. For our purposes, we have ours in a DNA RNA shielding buffer, which is just as effective. Okay, so we've added our samples into our two tubes, 1, 2, and 3. And these are still frozen, so we're going to wait on these to defrost. But while we're waiting on that, we will start to prepare our other tubes. Now, this part is the most extensive, the filtering part is the most extensive for this type of extraction, because you have a lot of other thick material that you want to remove. So when we start, we're going to add our fecals or soil samples into this bashing beads. So basically these are going to be vortexed, and it's going to bash up the material. So when we extract the material after they've been bashed and then vortexed, we're going to put them into this filter first, and this is the IIIF or 3F, I don't know, 3F filter. You can see. So one of the important things to remember with this is you're not going to throw away the collection tubes, because the material you don't want is going to be stuck in this filter, and what you do want is going to be left in this collection tube. So when you're labeling these, be sure to label these tubes. The next tubes are going to be the Zymo Spin IC tubes. Now, this is going to be different because what you don't want is going to end up in the collection tubes, and what you do want is going to be stuck in the filter. We're also going to create one set of tubes that we want to keep away from contaminants. It's one of the reasons why this biohood is very important to have and to use. So our last step, when we elute this, we're going to want to move these into these tubes when we elute, because the elution we're going to transfer to another step to prep to basically filter the eluted material. There's a lot of various compounds that you find in these kind of soil samples that might not get completely filtered out, or might get filtered out in the elution that you want to further process. Now, that's normally not the case if you're extracting DNA from blood or from urine or from saliva or from a series of other different sources. That's really unique, more unique with, oh wait a minute, what am I doing? I only need 3. It's really unique with these fecal soil samples. So I'm going to take this and put this in a drawer till we need it. Now these are called the Zymo Spin III-HRC filters, or the 3 HRC filters, however you want to say it. Oh, and just FYI, we have not been sponsored by Zymo Research, but I think some, YouTube University has come a long way, and I think these kinds of videos will be very beneficial to, sometimes you're just up all night reading, technical manuals, and you just need an easy button sometimes. And sometimes, just seeing it done once, even if you're a really experienced molecular biologist, you just need to see it done at least one time just to feel more comfortable. So with these PrEP filters, as I was saying earlier, this is going to be what we use to further process our eluted DNA. And this is much later on, so we'll go through each step. And last but not least, we need a column for our bashing bead sand thingies. This is going to be the first step. This process uses a lot of these racks, these test tube racks, so be sure to have like 5 or 6. 6 just for good measure. This is my favorite part. It's definitely labeling test tubes. And for this one, this is going to be the longest. So real quick, let's start here. As I was saying before, we can label the top just for easier identification, but we really need to label the side of these tubes for this one. For our experiment, we're basically looking at we're quantifying the amounts of our target sequence in the sample. So your experiment might be a little bit different in what you're doing. So we're doing qPCR with ours. Oh, and I almost forgot. We also need another rack to label just 1.5 microliter tubes for our finalized finished product. Okay, so this is going to be for our finished product. We'll put this away till when we need it later. And if you remember these from earlier, we forgot to label them. Alright, so we're going to go put these back away. As we're getting awfully cluttered in here. And let's go ahead and label these now and get this process started, because these have to vortex for about 40 to 50 minutes. I like to do 50 minutes. Actually, I lied. I set my timer in 51 minutes because every time I ask Siri to set my timer for 50 minutes, she thinks that I say 15 minutes. So if Apple's watching, please fix that. So now let's go ahead and, so these are 3 samples, so we're not going to be spiking anything into those. And this is our control, so we're not spiking anything into that. Just into these other tubes. And this is for our experiment. Like I said earlier, your experiment might be different. For this, we will be spiking in 100 microliters of each of our values. So we're going to be adding the whole sample to each of the bashing bead buffer or bashing bead tubes. Okay, so now these are going to be ready for us to vortex, and that will be the next part. Okay, so now we're just going to put these on here. And we will vortex these at 9,000 RPM. All right, and now we just let that vortex for 50 minutes. Hey Siri, set timer for 50 minutes.
15 minutes, counting down.
See? Proof. Hey Siri, set timer for 51 minutes.
There's already a 15-minute timer. Replace it. 51 minutes, counting down.
Okay, so while that is vortexing, we will go ahead and label the rest of our tubes. Ready? Okay, so now we have all of our tubes labeled, and we will let the Vortex Genie 2 finish vortexing, and then we will start the next step of our extraction. So now that our vortexing is finished, we will centrifuge these bashing bead tubes for 8,000 Gs for 1 minute. Okay, next for this part, we're going to take just 500 microliters of the supernatant off the top. I like to do this at this point right here, so we don't disturb the stuff at the bottom too much. So we want as little sand as possible, although it's going to be filtered out anyway. And those who have experience in running a mycology lab, you know that the contamination is, especially with trichoderma, it's really other fungal species that are in the air that you really have to worry about. And I think if you're doing a metagenomic study on the soil which the mycelium was growing in, you would probably want to do this part under a biohood, or at least a laminar flow hood. So now we're going to centrifuge at the last step. You want to centrifuge at 8 to 10,000. Sometimes 10,000 is better. But at this step with the filter, we're going to centrifuge this at 8,000 G. So the filters we don't want. And so we want to save this fluid here, and that's what we're going to add our lysis buffer to. So if you remember earlier, we were able to mark these on the side. So if I take this and throw it away, I know which tube is which. And now we're going to move over to the hood. So now we're going to add 1,200 microliters of genomic lysis buffer to this filtrate. So here we go. And if you remember in the beginning, this is the one that we added this stuff to. Mercapita ethanol. So our pipette only goes up to 1,000, so we're going to do 600 microliters twice, since we need 100. Now, normally if you're a beginner, you want to change out your pipette tip each time, but pro tip, if you can level it so it's right above, you can save a little money on tips. But you definitely don't want to dip this in and then dip it back into here, as then you'll cross-contaminate. Now this genomic lysis buffer, once it's in there, it'll really deactivate any kind of live bacteria or microorganisms that might be still alive. This is the end of the line for them. So the instructions say to pipette up and down 10 times to mix it. We'll do it a few times. I found that's a little bit more than what we normally need. But we're going to transfer it over into this. Now, these tubes can only hold up to about 800, 900 microliters, so we have to do this in 2 steps, which is a little bit of a pain, but since 1600 is I think the total is around 1600, yeah, so and remember, we're transferring this into the IC spin column. So we'll just vortex it. I mean, pipette it. Pipette to vortex. There we go. This here. So we should have around 700 to 800. Yeah, about 800 exactly. We can put a few more microliters. So I will adjust this from 800 to 825. These can hold about 900, but I don't like to go over 850. If you're doing more than 12 samples with this extraction, I would recommend clearing your whole day. Because out of all the different extraction types, this is definitely the longest. My sleeve almost grabbed one of these. So to be careful, I'll move this around. So this one's very thick, and because it's so thick, I know it's going to create a problem when we go to filter it. When we spin this down, it's going to really get stuck in the filter, and that's my biggest complaint. You can increase the speed of these, so for this next step it says to do it at 10,000 G for 1 minute. That's definitely not going to be enough. So I've done these up to 16,000 G. So we're gonna do 14,000 G for 3 minutes and see what happens, and see if it's still stuck. We'll check it, and if it's still stuck, we'll do it a little bit longer, maybe a higher speed. But just from experience, I can tell you that's what's going to happen. It's always good to make sure to keep track, so we're going to 10 to 2.5. Okay. We're going to stop this and move this over to the centrifuge so we can see what happens. If it gets stuck, we don't know. We're going to find out. Okay, so this time we'll do 14,000 G for 3 minutes. Okay, let's see. Exciting. Let's see if it got stuck. As you can see, it's stuck in there. So what we're going to do is, so that was 3 minutes at 14,000. So we're going to do it for another 2 minutes, and next time we'll put it at 14.5 for 3 minutes and see if that does it. Okay, so let's see if that did the trick. Yep, there's still a little bit in there. Let's try. This has happened before. Let's try higher speed. We'll try 15G. No, let's do 16G. I don't want to be here forever. Just for a minute. Okay, let's check it again. That's better. So we're going to dispose of materials. So everything that we want is going to be in the filter. So okay, so now that these are empty, we're going to put the rest in, the second half, and just be sure that you put the same positions into the same tubes. So now we're going to go vortex it the same as before. For the rest of the vortexing procedures, we're not going to bring the camera back over by the vortexer, but I think you guys get the gist. It's just a kind of a pain to move the camera. Like and subscribe to our channel, and maybe one day we can afford to buy a second camera. So, just for the record, this time I am doing it for 6,500 Gs for 2 minutes. So, we'll see if that works. And let's see the next step. So 200 microliters of DNA pre-wash buffer. So that is from the kit, and we just need to mark our date that we've opened this. Just to show you guys, it looks like that did it. So 6,500 G at 2 minutes was the golden number. If you're ever worried about not knowing how much to set it on, just follow the protocol. But if you do have stuff stuck in the filter, you need to try to increase it, both in the speed or the G-force that you're using, and also the time. So now I'm just going to go empty these out real quick. Alright, so 200 microliters of pre-wash buffer. We could use 1,000 microliter for this. I like the pipette tips better than for our 200. So once again, pro tip, we're going to reuse the same pipette tip. Now, if you're new at doing lab work and pipetting in general, I would recommend switching your tips each time. It really takes years of experience to have the kind of aim and steady hand to do it this way. Of course, this way is faster, and you save a little money on pipette tips. But if you do it wrong, you run a risk of cross-contamination. So sometimes there's, depending on the kind of extraction you're doing, there's two wash steps, but it looks like with this one, there's just the pre-wash and the post-wash. One time each. So we're going ahead and centrifuge this. So technically, you're supposed to do 10,000 Gs all throughout, and we increased it earlier because there was stuff stuck in the filter. Now, this pre-wash buffer will go through a lot easier. So we're not going to increase it as high as before, but we're not going to keep it down at 10,000. It'll still kind of get stuck. From my own experience, we're gonna do 2 minutes at 13,000 G. So we're going to prepare for the next step, which will be 500 microliters of the G DNA wash buffer. Well, I decided to do 14,000 G for 2 minutes, just because I want to get the filter membrane a little bit more dry before, make sure it's completely dry before we elute it in the next step. So if you remember our tubes that we had before, that we put in the drawer, we're going to get those out now. And we will be eluting it at 100 microliters. And you might be asking yourself, what is this for? Well, wait for it. Some of you might be asking, what does all this have to do with mycology? We're getting there. But this is my day position. So by day, I do molecular oncology by day, and by night, I am a mycologist. And the weekends. So the purposes of what this is for is completely secret, so sorry. Alright, so these are the tubes we're going to be eluting into. Eventually I want to do some more videos, and we're going to be going into molecular mycology. So this is all the junk we don't want. And now we are going to elute our DNA from our IC spin column. Now, if you remember earlier, I marked these, and that's because we're going to transfer the elution over into these columns, which are the, hold on. HRC, or 3 HRC. So I've definitely been very interested in, you know, some of the ways that cruciferous vegetables, you know, there's the chemical sulfuranes in them, which are activated by mysonerase into another compound. See it on the screen now. I can't remember all of these, but basically that compound is a powerful NRF activator, which has to do with over 200 different genes which help suppress tumors and fight cancer. And you're seeing, you know, there's similar lines of research with some of these medicinal mushrooms, like turkey tail. I mean, really anywhere that you see antioxidants, you see some potential benefit there. If you think about what antioxidants do for, if you're diabetic and your blood sugar is too high, then basically all of the sugar molecules are creating oxygen radicals, and that's creating oxidative damage, you know, in your blood vessels and on the cellular level, and that's one of the things that can cause diabetic neuropathy. And so these antioxidants are also very, very beneficial to really anyone, but especially if you have diabetes or one of these known diseases, and also just preventing disease and slowing down the aging process in general. Oxidative damage, you know, damages your DNA. The easier you can make it on the energy levels that it takes to replicate your DNA, the less stuff that your body has to do, and the more help that you give it, really the more you can potentially slow down the aging process. So that's something else that's kind of interesting. So for this part right here, I like to wait 10 minutes. It's more than, so in the protocol usually it's 3 minutes, but there's no difference between, the only difference between 5 minutes and 10 minutes is, it's a small difference. You won't get any more after 10 minutes. So if you let it sit for 10 minutes or 30 minutes, you're really getting the same concentration. The reason I like to maximize it, let's say sometimes you have a lot of tubes, and if you only wait 5 minutes and you centrifuge half of it at 5 minutes for a minute, it'll take a few minutes for you to centrifuge the next half. And by the time you centrifuge the next half, they will have incubated for 10 minutes. So by that point, it's going to throw your concentration off just ever so slightly. Really, the biggest difference is between 1 minute and 3 minutes. After 3 minutes, the rate really declines of how much your concentration changes, but I found 10 minutes just to be the perfect number. Also, while we're waiting for that, for the 10 minutes, we can go ahead and prepare this. So we can let this sit for 10 minutes, 15 minutes, it's all the same. So first we need to prep this. So we're going to put 600 microliters of the prep solution in here, and we're going to centrifuge it at 8,000 G. And all this does is process the sand. We're going to throw away the prep buffer, and after that, hold on one second. Remember this from earlier, these are our final tubes. So we're going to run the prep solution through, and then we're going to put this in our tubes. We're going to then centrifuge this. I hope your eyes aren't crossed. After we centrifuge this, we're going to take the elution out of the bottom, and then we're going to put it into the processed thing here. So you'll see. This is definitely one of the more complicated extraction processes. And don't forget to like and subscribe. We will be producing lots of content now that we have this camera. This camera is actually a Netflix approved camera. I almost kind of wish I had bought 2 cheaper ones. Instead of one expensive one. And we will be having lots of content come out. All sorts of subjects related to science, molecular biology, molecular oncology, and of course our favorite subject, mycology. In general, we have a large, well, I wouldn't say large, it's small right now, but we're growing, trying to grow. It's a mushroom growing operation. We'll also be doing breeding experiments, kind of what we've learned about how to prevent contamination, our quality control processes. You know, I absolutely love it here at this lab, and I love my coworkers here, and everyone's really great. But if it was up to me, I would probably be working on my mycology stuff full time. Just because, you know, well, it's my own business, and that's really a lifelong dream, you know. It's just, you know, you pick and choose your poisons. I guess I could have, you know, finished my PhD, you know, for another, you know, 3 to 6 years in school, and then I could have, I don't know, begged for research grants, and, you know, I don't know. We all have our routes to get to where we want to go. Mine was starting a business. That's definitely one way to do it. So now we will centrifuge these at 8,000 G for 3 minutes. Sometimes when I get really nervous, I like to smell my fingers and stick in my armpits. Superstar. Okay, is everything still on the camera screen? Okay, good. People have been bugging me for a long time to get into content creation. I don't know. Okay. Alright, so we have a prep buffer. We don't need this anymore. So these are going into our final tubes. So we are going to centrifuge our elution right now. And we're going to do this at 10,000 G for 1 minute. So I decided, since that one tube still has the filter a little bit clammed up, I'm going to do it for 16,000 G for 1 minute. So now, if you can see, we have our eluted DNA in the bottom, and so we're going to transfer this into these filters. Come on, AutoZoom, don't fail me now. There we go. So we don't need our filter anymore. And we just want our elution here on the bottom. Like so. Now we just got to do all the rest. Alrighty then. Okay, so now we just need to centrifuge this at 16,000 G for 3 minutes, and after that we'll have our sample. And I like to put mine in the -20. You could do -80. They'll stay okay at room temperature for a little while, or even at 4° in the fridge for a little while. And then either the same day or the next day, I will go in and process a qPCR for these. But let's go ahead and centrifuge them. So for this part, it's kind of hard to see, but when you put these in here, you want these edge pieces to be facing. So since it's spinning this way, the force is going to push this way. So if they're turned the other way, the tops will snap off. So I just kind of wanted to point that out. Come on, auto zoom. Come on, you can do it. There we go. So as you can see, that's our elution, and this is our final product. And we have our DNA from our fecal or soil sample. Soil sample collection can be very useful. One thing I've noticed in just studying mycology in general is some of the strains that you get from the wild are way more resistant to contamination than the strains that you find or what you can get online. So when you order a liquid culture syringe, you know, it seems like they've been isolated in these labs for so long, they're losing some of their potential to naturally fight off infections. So why does that happen? And one thing that you could do is you can analyze the soil or the region where the mycelium is growing, and you could take a sample of that soil or kind of the rotted wood material and just extract the DNA and run a metagenomic study. That's one thing you can do. First, one of the important things you're going to need to do is create a standard. So, or you can also buy standards, so you're going to want to know exactly what is in the soil sample. So usually if you have a target genome that you're looking for, it depends on how you're setting up your experiment. In this case, we have a target genome, and so we've created a standard curve.